Thank you. So welcome everyone, good afternoon. So what I will be presenting now is the first real truly global method to design RNAs and target some specific nucleotide distribution. And I will show how combined with local research that's resulting in a Glocal method, we can have pretty good results. So first of all, what are we talking about when we say design? Because design is still not a really well-defined field. What we are saying when we say design is really the inverse folding problem. So we have a specific secondary structure as the erpin here, and we try to find a set of nucleotides or a sequence that is going to have as minimal free energy this specific structure. And we have no proof of complexity for it, but we are convinced that it's a hard problem. So it's well worthy of our time and effort because there's no easy solution yet. And why we want to do that, and this is a really, really neat example, I think. And the trick here is that we have this trans RNA in purple on the top, and we would like to see when it is being expressed in the cell. So one thing that you can do is take a GFP mRNA and add a prefix to it to make sure with the prefix with the GFP, when they're alone, they're going to fall in this really nice helix, thus impeding the translation of the GFP. But when it's in presence of the trans RNA, it's going to open and allowing the GFP mRNA to be available for translation. And this requires a lot of different things. You need first to have this erpin shape when it's alone, but you don't want it to be too stable. It must be just unstable enough to open when we have the right trans RNA here in purple. So this is why we want to control in particular the GC distribution. So RNA structures, thank you for the really good introduction first about structures and energy. Of course, they're really in three-dimensional structure are pretty complex and twisted. So we only look at the secondary structure, which is simply a contact map between nucleotides interacting together. And what we're going to use as an energy model is the simplest of one, which is the basis of the Turner energy model. We're only going to use the stacked energy, the stacked base pair. So if we have an erpin, the energy is the sum of the stacks, and we can just go through all the stacks one after the other, look into our table, and compute them. Of course, the problem arises when you have, as here in the last base pair, AA, which is a non-canonical one. For those, we don't have experimentally computed energy, so we just use a parameter beta, which can be infinity if we want to completely impede them, or 15 if you like it, which we use from time to time, but it depends on your applications. So why local methods for now have a problem is if we look at the evolutionary landscape here, this is the space of sequences where every intersection is a sequence, and it should have 3 end edges going out of it to all one pointwise mutation neighbors. And if you start pretty much randomly into it and you want to go into the wells, which are probably the sequences that have the best affinity for a specific structure, going around the hills can take exponential time. So what people have been doing now in local design is kind of a divide and conquer approach in a lot of different tools. So if we have a really simple secondary structure at this one, which is simply two erpins concatenated one after the other, what you can see is, oh, this is the same erpin, in fact, twice, which is concatenated. So let's be simple. Designing for one small erpin a sequence that's going to fold into it is relatively easy. It can be done quite fast. So you can just try to compute it at first. And then the logic idea would be, well, we can just concatenate the two sequences together. We might have the concatenation of structures. And in practice, it works relatively well. But of course, you have a lot of cases. In theory, there's absolutely no certificate that when you do that, you're going to have the required structure. And in particular, in that case, if you do it, you will just have a huge erpin, which is completely different from the target at the basis. Thus the need for a global sampling. And those are the only equations in all the talks, and I'm not even going to talk about them. So what we are using is here a simple Boltzmann distribution, and it's a bit different from what we have seen before, because here we have only one structure, and we look at the set of all possible sequences, and what is the relative energy for this specific structure. And since we have a simple stacking energy model, which is additive, we can use dynamic programming equation to compute them really, really efficiently. And the idea is, if one sequence, as the pink or green one, has a better affinity for a specific structure, If we then look at the set of structure, we're going to hope that this structure is overly represented. Of course, affinity is not the same thing as specificity. And if we look in particular maybe at this big sequence, maybe it's just that it has a really low energy. And when we look this time, this is the Boltzmann set of structures for this specific sequence. Although the sequence has a high affinity compared to other sequences for the target structure, you can see other structures which by themselves are really different and have a better weight in the structure set. At the opposite, if we look at a smaller energy in the sequence ensemble, well, its structure ensemble is just much smaller because due to its nucleotide composition, its global energy is smaller. And in that case in particular, well, this target structure is the most representative of the Boltzmann set. So this is a problem, but in general, it is a viable alternative to the local search and genetic algorithm, and it works relatively well, so it can be used later on. So it's a good idea. So this doesn't come from nowhere. It was developed by Levin and al. In 2012, what they call RNA Design, which was a proof of concept of global sampling could work. But there's a lot of differences between those, which are really important. First of all, RNA Design is a seeded method, while Incarnation is not, which means that Incarnation, which is the name of our software, just looks at the total set of sequences from the start. There's no bias from any starting point. Well, if you explore the mutant space as RNA Design does, you always have a slight bias. Of course, if you compute mutants that are far enough from the seed, then you compute effectively the whole space. If you do that with RNA Design, you have a big O of N5 complexity. While with our method, we have a linear dynamic programming algorithm allowing us to compute everything at once. Of course, the higher complexity comes with some advantages. They have a much more complex energy model, almost a full Turner energy model, while we're only considering stack base pair, which is a really simple basic model, but it works well enough for now. And there's another nice thing that we've done with Incarnation is that we can simply add sequence constraints inside. So if you want to have some stretch of nucleotides in particular or some position, you can just add them into the software directly, and it's even going to speed up the time it's needed to compute because we don't have to look at alternative. And just now, which has been accepted to ACM BCB, there's another paper by Ponti and al. About how they can use language theory to also in linear time add a lot of sequence constraints really easily to this type of application. So you can go really further than just blocking some position or allowing a subset of nucleotides to them. So how all the magic works, this has been inspired by a talk I presented at RECOM, so you might recognize the next 4 slides. The trick is really that the structure is fixed. We always have a target, so what we want to compute is the full sequence space. So in that case, we already know the structure, we know what are the nucleotides I and B, and we're interested in the partition function of all possible sequences in this purple region. And we can divide that in 3 simple cases. The first case is simply that the first position is unpaired. That case, since we only consider stacked energy, there's no even energy contribution to it, and we only have to compute, again, the partition function in the purple set. The second case is when we know that there's a base pair between the first and the last position, and there is a base pair between the 2 external nucleotides too. Again, we already know what A and B are. This is set. The structure is set, so we only have to add over the 16 different pairs of nucleotides A' and B' for the energy, and compute the partition function again of the purple inside. The most general case, the last one, is when we simply have the first position paired somewhere else in between. Again, no energy contribution directly from that, and we only have to compute those 2 partition functions, one inside the base pair and the one at the exterior of it. So we have only 3 simple cases. And what's really important to remember is that we already know the structure, so we already know in which cases we're going to fall, so we only have to look in our table, follow the path, and this can be done in linear time really efficiently. So this is computing the energy. Now, how do we backtrack? This is a simple global stochastic backtrack, really useful and convenient. So this time we already have for this structure all the energy. So when we look at the first position, we're going to do for all the 4 possible nucleotides at this first position, the weight for picking it up randomly is going to be the partition function of the purple inside, given the fact that we know which nucleotide this is going to be. So since this doesn't add energy, in fact, we just pick one randomly, the second is more interesting because the fact that the first and last nucleotide positions are set is going to influence the partition function of the purple because you have a stacking here. And all the different, for all pairs, we're going to have different values, and those values of the partition function are going to be the weight for the random sampling of those 2 nucleotides. And you just go forward inside computing 1 or 2 nucleotides at a time. If you have hairpins, you just can do it one and after the other. And again, this is really fast and efficient because at every time we just do a constant number of operations to go through all the different positions. And of course, we use memoization, so we remember all those values. We don't have to compute them again. It's just in a table, really, really fast and efficient. So, of course, there's a problem, and this is the GC bias. Here in the left, what we have, this red curve, is in fact the distribution of GC content when we sample 1,000 different sequences using our global method. In yellow, what you have is the average over all RFAM sequences, which is 48.8%, and we are around 64. On the right, this is simply the distribution for the different local design softwares as NewPack, InfoRNA, RNA Inverse, and Frankenstein. And you can see that they are all shifted to the higher values of GC content. And this is expected because usually what you will try to do is minimize the energy, and GC base pair have a lower energy, so you would expect to have in the sequences a higher number of GC content inside. But it's a problem because if you recall the first example, if it has a lower energy, it's more strongly bound together, and if you want for some application for it to be able to open, then it's really important to be able to target sequences which have in this range where for now nothing is there. Thankfully, there is this absolutely wonderful trick that has been developed by Vladimir and Ponzi, presented at RICOM in 2011, if I recall well. Well, the trick is not simply, because it was not simple nor the proof, but for every recursion, what you can do is count the number of GC that you have at the position that you're looking for at every step, and find a suitable value of X that is going to shift the distribution of sampling. And that's really important in an unbiased way in regards to your prior distribution over what is the effective range. So this might look complicated, but there is this time a really simple approach to do that, and it's simply a bisection scheme. So we can start with a value of X, which is 0.25 for every possible nucleotide, and this is the first graph that we had. So when we just leave everything be, what we have is around 65% of GC content in sample sequences. And if we target 15% plus or minus 5%, we can see that at first round with that, we have simply 0 samples that fall into our target. So what we do is simply we divide X by 2, and this shifts the distribution slightly to the left. And that time we have some samples, 100 over, if I recall well, 1,000, which are exactly in the right region. And we can continue doing that again and again and moving in between. And at some point, it's going to stabilize. It looks like it might take a long time, but in fact, there's a proof that exists that it can take at most linear time to be exactly where you want to be. And in practice, after 5 or 6 jumps, you will get already will have enough samples, that even more samples that you want to have. And again, what is really important is that you only condition the prior distribution with the amount of GC, but for all the sequences that are sampled in this region, the distribution of sampling is the prior one, which depends on the Boltzmann ensemble of sequences for your target structure. So this is still unbiased in regards to the interesting distribution. So does it work? Not really. If we look at the samples, of course, as I said, we have no guarantee that they're going to fold when we compute the full minimum free energy using RNA fold into the desired structure. And as we can see, this is the stack density, or the number of base pairs. And more base pair we have, more energy term we have. So we can have roughly 80% at the extremes of sample sequences that have the right MFE, minimal free energy structure. But for most of them, that doesn't happen. So why is that? If we look first at local versus global versus glocal. So the local technique is you start randomly, you try to move around your sequence space and try to find those wells, which can take a lot of time. With our global technique, which is really fast, what you do, if in fact, is you sample points which are always pretty close to the deeper wells where you have, but you don't have guarantee that you're going to hit the lower point at every time. So we had the idea that we are only playing with positions that are involved in a base pair. So what happened if we use our sample sequences as seed with local methods that accept seed, and we only allow unpaired position to be mutated? And this turns into a glocal approach, where effectively what you do is that you pick your seeds directly already close from the really important and desired location, and the local part will just move a little bit around and try to find the best solution. And when you do that, it's much more efficient. Here in the bottom part, we have the success rate, again in function of the stack density, which is the same thing, percent of correct minimal free energy structures for RNA inverse and for RNA NewPack and RNA SSD. And we can see that for NewPack and RNA inverse, they do worse than us, at least for lower percent of lower stack density. Infor-RNA does really well, but when you look at them, they have roughly around 80% of GC content, so this is not desirable. And RNA SSD, although does really well. So we were interested in comparing now with RNA SSD, how diverse are the set of sequences. So this is something that is really important because as it's written in RNA fold documentation, the nature is often wrong. It doesn't fold as it's supposed to be when you use our tools. So we are interested for practical application to sample first a lot of sequences that we are pretty sure are going to have the right structure, but we want those sequences to be as diverse as possible. So we want to have the lowest possible sequence identity between them. And when you use our tool Incarnation plus RNA inverse around this 10% of GC content, what we can see is that we have a slightly better sequence identity than RNA SSD. And around 30% of GC content, it's pretty mixed in between those two. So is there really an advantage of using our tool? Well, although we are pipelining it through RNA inverse, which is a cubic time algorithm, RNA SSD is also a cubic time algorithm, but the difference in time is just incredible. Here we're only going to lengths up to 90 nucleotides, and we're taking less than half a second to compute any given sequence, while RNA SSD is exploding really fast. And yes, you can notice this sharp drop at the end, but this is in fact underlining another huge problem that we have with local approaches, where the secondary structure is going to have a huge influence on how easy or hard it is to design specific sequences for that. While using our global technique, this is not really relevant because we just parse the structure once, then the sequence once, and we already have our samples, and it doesn't have an influence on time or difficulty. And then the RNA inverse step is thankfully not taking much more time, and it's almost, again, a linear trend, although RNA inverse by itself is a cubic time algorithm. So we were pretty happy with the results of this Glocal technique altogether. But of course, this is just the beginning. What we would like to do now is first implement a more complex energy model, maybe go to n to the square for Incarnation. Yes, it will take some more time, but it's probably still pretty efficient, and we may have really better results. This is something we have to check. And using seeded, local seeded method it seems a pretty good idea. And we only use RNA inverse, which is with the Vienna package, really well known, really useful, but it's also one of the most simple that we have. There's a bunch of others that we would like to try, and maybe we can get even better accuracy using those ones. Finally, probably the hardest part in a theoretical lab is convince people to make those molecules in lab, especially large one. If you want to design some funky RNA structures or do something, please come talk to us. We will be really, really happy to help you and see and be able to really benchmark those tools finally. So thank you very much. I would like to thank in particular my co-authors and supervisor Jerome Valdishpool and Jan Ponti, Flying Spaghetti Monster for its inspiration to innovate, and my way too many funding agencies and people. Thank you very much.
Yeah, thank you. Questions? Yes.
How could experimental validation look like?
It depends on what application you want to have.
Maybe because you have known the structures, but I wonder how to validate them by the environment.
Well, I think that maybe the first example was a pretty good one. If you want to, if you have a specific sequence as a GFP and you want to be able to fold it into an helix.
Exactly.
Yeah.
So the structure you showed is known, the first one, because then the question is, when you have done all your calculations, how many would fit to this first sample?
We don't know. We don't really know, I think.
But since you have done the analysis, as I understand, you could now...
Oh, we didn't do the first experiment. That figures was from another paper and explained really well the point that
it's... know the structure of the first one.
Well, a simple helix is simply an helix, but no, I don't know the structure from it. But
you could use your system to...
yeah, we could use it.
And if you do this, then you could check all your other structures.
Absolutely, yes.
How closely you can. And then we have the probability of distribution.
Yes, and we would hope it works well, but we don't know yet.
Okay, another one.
Do you have any plans to support knotted topologies at any point in the future?
To support what, sorry?
Not like pseudoknots and things?
Well, usually in all RNA talks, if pseudoknot is not enlarged, it's because we try to hide the fact that we don't talk about them. Well, if you use pseudoknots, if you use really simple pseudoknots, the complexity doesn't increase that much. We can use a lot of really neat things like multi-level folding or stuff like that, as has been developed by Sato et al. With integer programming. I mean, I think everyone wants to work with pseudoknot, but we didn't have a good idea. If you do, please come talk to us. We'll try to do something with that. But I'm pretty sure it's really important. It's just that it's really hard to do for now. Thank you.
So I think there are no more questions. So this closes the session. Thank you again. There's half an hour.